Dear students, welcome back to the class. Today's topic of discussion is acetylcholine and cholinereceptors. As in the last class, we have discussed about different types of neurotransmitters and the functions of neurotransmitters. So today, acetylcholine, among one of the excitatory neurotransmitter, we are going to discuss about its topic and its classification. So let's once again start with this topic in details with the following presentation slides. Let's quickly look into the contents. Here I have included the points introduction, synthesis, storage and release of acetylcholine, synthesis and release of acetylcholine, cholinereceptors and subtypes, muscarinic receptors, nicotinic receptors and the reference of the entire presentation. So this is the introduction about what is acetylcholine. Acetylcholine is basically a most abundant neurotransmitter that is present in our body. It is present both the central nervous system and the peripheral nervous system. In the central nervous system, the cholinergic projections from the basal forebrain to the cerebral cortex and hippocampus support the cognitive functions for those target areas. While in the PNS, the acetylcholine activates the muscles and is a major neurotransmitter in the autonomic nervous system. So, acetylcholine has its major function both in the CNS and PNS. In the CNS, it helps in the cognitive projections, and in the PNS, it is acting as a neurotransmitter. Here, you can see that it is the first neurotransmitter scientists have discovered, as well as the most abundant neurotransmitter in our body, as I said previously. So now this is a chemical messenger which is released from a neuron to a nerve cell that sends a signal to another neuron across a synapse. And this is the structure of acetylcholine. Next is the synthesis, storage and release of acetylcholine. Synthesis of acetylcholine. It is synthesized at the cholinergic nerve endings. So here you can see that this is the synaptic cleft. From here, the choline and the sodium ion that is entering the postsynaptic neuron. So here, what happens? This choline in presence of acetyl-CoA, which is present in mitochondria, in presence of choline acetyl transferase, synthesize the acetylcholine. Now, you may have a question that where is this choline coming from? This is coming from as a recycled product from acetylcholine degradation, phosphatidylcholine breakdown, and dietary supplements. So these are the source of choline, or you can say these are the sources of choline for the synthesis of acetylcholine. And this acetylcholine is mainly synthesized at the cholinergic nerve endings. Next, we come to the storage. After synthesis, acetylcholine is stored in the presynaptic vesicle. In this presynaptic vesicle, what is happening? The uptake of acetylcholine into storage vesicles occurs through an energy-dependent pump. So this is the energy-dependent pump that acidifies the vesicle. And this acidified vesicle then uses a vesicular ACH transporter, this vesicular ACH transporter, that is VACHT, to exchange the protons for acetylcholine molecules. So the storage is done in presynaptic vesicle, and the uptake of ACH or acetylcholine is dependent through a energy dependent pump, or occurs through a energy dependent pump. And the last one is the release of acetylcholine. Acetylcholine is released by the process of exocytosis. So exocytosis, what happens? If we start with the initiation process, that is, at first there is the action potential or nerve impulse generation, then calcium ion influx, migration to the synaptic vesicle, release of acetylcholine to synaptic cleft, binding to receptors on postsynaptic membrane, entry of Na plus depolarization of muscle membrane, contraction of muscle membrane, and ultimately release of acetylcholine by the process of exocytosis. Next, here, let's recapitulate with the synthesis process of acetylcholine. Here you can see acetyl-CoA production of acetylcholine, which is collected in the synaptic vesicle, released as a neurotransmitter in the synaptic cleft, then it is again received. And here let's collect the points that what are the steps are happening in the synthesis and recycling of acetylcholine. Step 1, you can see acetylcholine is made from choline and acetyl-CoA. Then step 2, that is in the synaptic cleft, acetylcholine is rapidly broken down by the enzyme acetylcholinesterase. This is the enzyme. Step 3, that is choline is transported back into the axon terminal by co-transport with sodium ion. And the last point, that is the recycling, that is recycled choline is used to make more acetylcholine. So this way we complete the synthesis, storage, release and recycling process of acetylcholine, the excitatory neurotransmitter. So now let's come to the receptors. Now, what are the name of the receptors? The name of the receptor is cholinereceptors, and this cholinereceptors is basically two types, or cholinereceptors are basically two types. Now you can correlate that acetylcholine, that's why cholinereceptors, and they are two types. The two major types are muscarinic and next is nicotinic. Muscarinic is divided into 5 major types, that is M1, M2, M3, M4 and M5, and nicotinic is divided into NM and NN receptors. Now for this concept of receptors, 3 things we need to study. First, the location of the receptors. Second, the function of the receptors. And third, the agonist and antagonists of the receptor. But for this presentation, we are only going to study about the location of the receptors, both muscarinic and nicotinic. So now what is muscarinic receptor? Muscarinic receptor is stimulated by muscarine. They are blocked by atropine and they are located in the autonomic effector cells in heart, blood vessels, smooth muscles and sweat glands. This is the general location of the entire muscarinic receptor, and in CNS, a subsidiary muscarinic receptor is also present in autonomic ganglia where it plays a modulatory role, and most importantly, muscarinic receptor is a G protein couple receptor. So these are the 5 key points of muscarinic receptor, and from this part you can even get multiple choice question such as dash is a type of G protein couple receptor. You can get options like nicotinic, muscarinic, adrenergic. So you have to tick on muscarinic receptors. So here is the location of muscarinic receptor with its subtypes. So they are basically 5 different types of muscarinic receptor. M1, which is located in salivary glands and enteric nerves. M2, that is heart and smooth muscle. M3, that is smooth muscle and salivary glands. M4, brain, lungs. And M5, that is brain substantia, that is in the lungs. So M4 and M5 is almost the same. So here I am going to highlight M1, M2 and M3. So for M1, I will tell that M1 plays a major role in mediating the gastric secretion, relaxation of lower esophageal sphincter, and in learning, memory and motor function. This is the function of M1. So it plays a major role in mediating gastric secretion, relaxation of lower esophageal sphincter, and in learning, memory, and motor function. Next is M2, that is, it helps in the cardiac muscarinic functions, predominantly M2 for mediating the vagal bradycardia. So what it is saying, the autoreceptors on cholinergic nerve endings are also of M2 subtypes, that is, smooth muscles which express some M2 receptors as well, which like M3 mediate contraction. So here you can see the muscular contraction is the main function of the M2 muscarinic receptor. Next is the M3, that is a visceral smooth muscle contraction, and glandular secretions are elicited through M3 receptors, which also mediate vasodilation through endothelium dependent relaxing factor release. So we can say that together with M2 and M3 mediate most of the well recognized muscarinic actions, including the contraction of lower esophageal sphincter. So this way, the various types of muscarinic receptor subtypes with their location and function has been discussed. Next is the nicotinic receptor. Nicotinic acetylcholine receptor, or in short you can write NACHRS, are receptors which are polypeptides that respond to the neurotransmitter acetylcholine. And these nicotinic receptors respond to drugs such as agonist nicotine. They are found in the central and peripheral nervous system, and at neuromuscular junction they are the primary receptor in muscle for motor nerve muscle communication that controls the muscle contraction. In peripheral nervous system, they transmit the outgoing signals from the presynaptic to the postsynaptic cell within the sympathetic and parasympathetic nervous system. And you know what is sympathetic, what is parasympathetic nervous system? And the second function is they are the receptors found on skeletal muscle that receives the acetylcholine released to signal for muscular contraction. So these are the overall functions of nicotinic receptors. In the immune system, or you can say the nicotinic acetylcholine receptors regulate the inflammatory processes and signal through a distinct intracellular pathway. So in insects, the cholinergic system is limited to central nervous system. So this is an added point that for insects, what happens? So now these are the two types of nicotinic receptors. See, nicotinic receptors, as I said earlier, it is NM receptor and NN receptor. See, NM, M is in the subscript, which means present in muscle. You can remember in this way the location of the receptors, and NN, which means nerves. You see, it is located skeletal muscle neuromuscular junction, and it is in the postganglionic cell body and dendrites. So in this way you can remember the location of the nicotinic receptor subtypes. So this is the reference of the images that I have shared. Thank you students for listening to the presentation. Hope it will help you in doing your assignments. For any doubts and queries, feel free to ask.